Hello and good morning. My name is James Wood. I'm an infectious disease veterinary epidemiologist based at the University of Cambridge, and I'm one of the co-directors of the UK International Coronavirus Network. And it's a great pleasure to be part of your meeting this morning and to be connecting, albeit indirectly, with my old friend Wanda Markotter, who I have not seen with this pandemic for far too long. I can give a few slides on wildlife biosurveillance and some of the challenges that I think that we have in relation to using wildlife surveillance data. And obviously an underlying thread thinking about wildlife biosurveillance is, can we detect the next pandemic at source? We all know that zoonoses are important for human diseases. A very high proportion of human infectious diseases come from animals originally, and a far greater proportion of those emerging infections come from animal sources. And it's interesting to note that of these, particularly more recently, at least 3/4 of these have a wildlife rather than domestic animal source, which is perhaps not surprising given the relatively small number of species that have been domesticated and the huge number of animal species that have not. I think you all know that emergence of zoonotic pathogens from wildlife is not something that occurs consistently over the world. This is work that was published by Kate Jones nearly 15 years ago now, which that demonstrates distinct hotspot areas for pathogen emergence from wildlife. It's very important to distinguish true effects from those of diagnostic or surveillance bias, and I think despite good efforts to try and moderate that in this paper, there's probably still some investigative bias in these hotspots, but nonetheless I'm sure that for some of the larger areas there are genuinely increased areas of risk which will relate to such things as a lot of contact between people and wildlife, having a lot of wildlife present in the area, and so on. These were emerging diseases that had been characterized. It has been estimated there are between 1 and 2 million other pathogens that are yet to be discovered that are circulating in different animal species, and perhaps for some of them between different animal species. So thinking about what we're trying to try and prevent, we're trying to prevent transmission of new diseases in humans. And I think that it's very important to note in the context of pandemics that is if an infection arrives and it transmits readily between humans, pretty much anywhere in the world it is likely to spread. And if it is spreading rapidly, then many infections, particularly RNA viruses, for reasons that I'm sure you all understand, they can evolve very rapidly. And I think SARS-CoV-2, the cause of the COVID-19 pandemic, has demonstrated very, very clearly how quickly a pathogen can adapt for greater transmissibility. We've seen a succession of different variants over the two to 3 year course of this pandemic now. The most obvious ones being Delta, Omicron, and Omicron BA2, and I'm sure, unfortunately, that we will see more of these. So the key question I think we need to ask is, can we detect and intervene effectively at source of a pandemic? Because when we think about how rapidly pathogens can transmit at the beginning of a pandemic, but they don't always, we need to think about really rapid intervention, typically with non-pharmaceutical measures. So we're talking about isolation, distancing, and so on. Can we and should we intervene at source where we can? And you will know, particularly in South Africa, that measures should be proportionate to what is being dealt with, both in one country and in any other country that might be trying to isolate itself from another. So let's think a little bit about how good our surveillance data are, because a key defense against disease emergence is early detection. Early detection requires surveillance, and that requires primary healthcare, laboratory capacity, and access to the laboratory capacity from primary healthcare, and obviously excellent communications. Another way of looking at it, which we don't often, but perhaps should more in the future, is can we sequence pathogens from lots of people who have contact with animals for early detection? Think about the infections that are likely to cause problems in humans. Or should we focus on pathogens within wildlife when we have these 1 to 2 million? So there is an advantage to thinking about sequencing pathogens from people who have developed clinical signs and perhaps have a fever, who have contact with animals, because that straight away cuts things down into pathogens that have, even if they don't transmit, but at least demonstrated the ability to replicate in humans. So in order to challenge our thinking, I want to talk about some human health-seeking behavioural studies that have been done. Because obviously humans have to seek medical treatment if they are to contribute to surveillance programs. I just want to really in one slide try and capture a 5-month ethnographic study that my colleague Dr. Fred Jeffcott undertook in central Ghana. Now this is a malaria hyperendemic rural region of Ghana. Ghana is said to have some of the best primary health care in much of sub-Saharan Africa, but like anywhere, it's not perfect. It's impossible to capture 5 months' work in one slide, but the really important take-home messages from this study, where village and town living were compared, where there were really different patterns of healthcare usage in the communities that were being studied by Dr. Jeffcott. And healthcare usage was influenced hugely by income, the signs of disease people had, and how severe the disease people had. What Dr. Jeffcott found was that in many of the clinics, there was inadequate record keeping and somewhat variable communication between doctors and their patients. With the lack of a really good consistent record keeping, and because this was a malaria hyperendemic region, there was frequent retreatment for malaria if people represented with fever at the same clinic, despite the fact that the first treatment hadn't worked. There was pretty much only one opportunity for a single diagnostic test, and that was a rapid test for malaria off the blood smear. And even that was not always used. And also you have to remember in these hyperendemic regions is that a lot of people will be parasitemic for malaria, even if their disease is not being caused by that parasite. With all of these things taken together, and I would encourage you to read the paper in full, unless there was widespread transmission of a disease with a new clinical presentation, there was little or no chance of detection of emerging infectious diseases in this area where primary healthcare, as I said, is relatively good compared to much of the continent. What about a pathogen that we know, fear, and study, and the narrative internationally is that we have good surveillance for Ebola? Now, thinking about Ebola, we know how big the detected outbreaks are because they are all reported, written up. And interestingly, through using fairly straightforward mathematical modelling approaches, we can predict how big the outbreaks should be, given how transmissible the infection is during some of the bigger outbreaks where it has been possible to study and quantify this. And from this, we can predict what the distribution of outbreak sizes should be, given how transmissible the infection is. So this is work that was done by Dr. Emma Glennon that was published 2 or 3 years ago. And she used data from the big West African outbreak, over 5 years ago now, to understand how well the infection spreads when it is spreading in a major epidemic. And so Emma used data from Guinea, from Sierra Leone, and also in green in the combined data set. And using that information and a really detailed analysis of the West African outbreak, Dr. Glennon was able to look at, in that real life setting, how much each case should transmit. And you can see the distribution of secondary cases from each reported case, diagnosed case. And you can see that that there is quite a long tail. You get a number of individuals that transmit quite a lot, to more than 7 to 10 people, but the vast majority either don't transmit or transmit only to 1 or possibly 2 people. So then, looking at the 3 different slides here, three different graphs from her paper in Plus Neglected Tropical Diseases. The coloured line on the left-hand side represents how big the outbreaks should be based on this transmissibility, and the grey shading around the hatched line represents the actual probability of detecting outbreaks of different size based on the published scientific literature and reports to WHO. And what you can see is once you get above 5 cases or so, that the likelihood of you finding this, reporting this outbreak, observing it, is very high, particularly once you've got above 10 cases. But actually, for single cases that do not transmit, the likelihood of detecting them is less than 10%. So the point being that we only detect Ebola when it transmits between people. We're not detecting single cases of Ebola, and this is a measure of the ability to investigate single causes of severe illness or death in West and Central Africa, where this most feared infection is currently located. Overall, there was a finding that at least half of all spillover events have failed to be reported since Ebola was first recognized. As I said, that the likelihood of you finding outbreaks for small outbreaks is less than 10% for single case spillover events. And that for me is very strong evidence of a need, if we want to try and detect pandemics at source or do anything about them, is that we must internationally invest in primary healthcare to protect the world, as well as providing better healthcare for populations at highest risk of zoonotic infections. So going back to the whole point of this exercise, thinking about transmission in humans, I think for acute infections, ones that are not transmitted over a long period, such as HIV, which really was 50 years between international emergence and its first transmission from non-human primates into humans. For acute infections, intervention at source is very unlikely to be effective. And I think we have to be realistic about this. And for me, this is why we logically should therefore intervene to try and prevent spillover. Think about really important measures, think about reducing biodiversity loss, challenging extractive industries, which can underpin a lot of the biodiversity loss and the destruction of pristine areas. I think very much about intermediate hosts, be they wildlife hosts or that are being farmed, or straightforward agricultural animals. Which needs on there, therefore, to consider what the role of wildlife farming is. So the first pandemic of SARS, now called SARS-CoV-1 in 2003, was driven by the farming and trade of live palm civets in South Asia. There was an extended epidemic of SARS in these traded palm civets, which caused multiple spillover events into humans, which repeatedly seeded SARS into the human population, where palm civets were acting as an intermediate host between humans and bats, horseshoe bats. The key question that probably we'll never be able to answer for sure without uncertainty is to ask whether there was a role for farm wildlife, which were common in the Hunan seafood market in Wuhan, in causing the onset of the SARS-CoV-2 pandemic. I suspect there was, but this is no more than a suspicion. It's something we cannot be sure about. So then the question is wildlife farming different to more conventional agriculture? Well, it's important to recognise the scale of wildlife farming in South Asia is massive, and it's large elsewhere too. In many cases, species that can be very effective, bridging or intermediate species for pathogens spreading between wildlife and humans, are the species that are being farmed. And when you move them around and mix them alive with other species, this is a potentially great way of spreading potentially very dangerous pathogens into the human population. That's not to say that agriculture, live animals in agriculture, movement in particular of live animals in farming is not still a massive risk. But I think that the mixing of wildlife species and so much of wildlife farming, when they're being traded alive, is something that does set it aside from conventional agriculture in terms of the scale of the risk. So then, really to finish, is there a role for massive investigation of pathogens in wildlife, thinking about metagenomic approaches to try and discover what infections are present in every species that we can sample ethically? It is, of course, invaluable to know what pathogens are present in different species of animals. But what are we going to do with the information? How will we really predict the next pathogen or pathogens that will cause a pandemic? And I draw your attention to the fact that we cannot accurately predict which influenza strain will cause the next seasonal flu outbreak. We make our vaccines in the Northern Hemisphere based on those viruses that are circulating the previous season in the Southern Hemisphere and vice versa. So that's really more of a rapid evaluation rather than a prediction. And remember that at the moment we don't have multiple cross-reactive effective vaccines. We don't have the technology. Well, we may have the technology that could produce them, but as yet we don't have a widespread evidence that we can produce these. And then if we were able to produce them, are we going to be able to stockpile them, or even stockpile vaccine seeds for these products for a pathogen that currently is not spreading in the human population? Who's going to pay for that? And given the challenges of resourcing primary health care at the moment, I think this is a really important question to ask when we're thinking about what the best way of spending money to prevent or mitigate the impact of the next pandemic. It's been a real pleasure to be here this morning as part of the UK International Coronavirus Network. It's fantastic to have this meeting to be held in association with UK-ICN. This is some contact information for the network, websites, Twitter and email potential, and I hope that many of you will be able to interact with the network over the course of your work in the next few years. Thank you very much.